Usually when we think about the future of healing or um, regenerative medicine, there's this expectation that it's going to involve transplanting whole living stem cells right into your body.
Right.
So
Yeah.
the general idea is essentially like replacing a broken part in a machine
Yeah.
with a brand new part.
Yeah, it's the classic, highly structural view of modern medicine.
Right.
The logic basically follows that if a tissue is failing, whether that's a joint, a heart muscle, or a severe wound, you need to put fresh, viable, living cells in there to physically rebuild it from the ground up.
But, today we're looking at research that suggests the future of medicine it isn't about transplanting new cells into your body at all.
No, not at all.
What if instead the secret lies in harvesting the microscopic care packages that those cells leave behind?
Yeah, that is exactly where the frontier of regenerative medicine is right now. We're moving away from the cell itself as the primary therapy
Wow.
and focusing entirely on the chemical messages it sends.
Welcome to this deep dive. Today, our mission is to explore a central, honestly mind-blowing premise found in a massive stack of scientific papers.
Oh yeah, there's so much data now.
Right, like we're talking comprehensive proteomic analyses, reviews from MDPI and Frontiers, and clinical studies on nanotheranostics.
It's a lot to go through.
It is. But we're looking at how the specific tissue origin of a stem cell completely alters the growth factor profile inside these microscopic care packages.
Exactly.
Basically, we're going to look at how pulling a stem cell from bone marrow versus fat tissue versus an umbilical cord turns these packages into highly specialized tools for totally different medical emergencies.
It's a profound shift, really, in how we understand cellular therapy. We're no longer treating all stem cells as, like, generic magic wands.
Right.
We're looking at how the neighborhood a cell comes from dictates the specific therapeutic toolkit it produces.
Okay, let's unpack this. Before we get into where these packages come from, I think we need to establish exactly what they are.
Yeah, good idea.
So these are called mesenchymal stem cell exosomes,
Right.
and they are defined as nanoscale extracellular vesicles. When we say nanoscale, we are talking about something roughly 30 to like 150 nanometers in diameter.
So tiny.
Yeah, so picture a single virus particle. That is how small these things are.
Incredibly tiny. And, historically, they were actually ignored.
Wait, really?
Yeah. For a long time, researchers thought these vesicles were basically cellular trash cans.
That's just garbage.
Exactly, just a way for the cell to throw out metabolic waste. But when they actually looked inside these incredibly small lipid bilayer membrane vesicles, they found something else entirely.
They've had a highly complex cargo, right?
Yes. An intricate mix of active proteins, lipids, messenger RNAs, and microRNAs.
Wow.
For years, we thought the main therapeutic value of a stem cell was its ability to physically turn into other types of cells, like a stem cell deciding to become a bone cell.
Sure, that's what I was pictured.
Right, but the data now overwhelmingly shows that their primary benefit comes from what we call the paracrine effect.
Okay, let me make sure I have the paracrine effect right. Basically, the transplanted stem cells aren't necessarily doing the manual labor of building new tissue themselves.
Right.
They act more like project managers.
Exactly.
They secrete these exosome packages into the surrounding microenvironment to tell your existing cells what to do.
Mm-hmm.
Like they send the instructions to build blood vessels, manage inflammation, fight off stress.
That captures it perfectly. They are the management, not the manual labor. And understanding this is where the advantages over traditional whole cell therapy become, honestly, absolutely staggering.
Because they aren't whole cells.
Exactly. Because these exosomes are not whole living cells, they cannot replicate on their own.
Which means they don't carry the risk of accidentally growing into a tumor.
Yes.
I know that has always been a massive hurdle and a huge fear with viable stem cell transplants. If a living cell goes rogue and keeps dividing, you have a major problem.
Right. I mean, a whole cell is alive and unpredictable. An exosome is just a biological container.
Okay, that makes sense.
It also doesn't possess the same complex surface markers as a whole cell, so the risk of your immune system attacking it and rejecting it is drastically lower.
Oh, that's huge.
And from a logistical standpoint, think about how difficult it is to keep a batch of whole human cells alive, sterile, and perfectly viable to be transplanted into a patient.
Yeah, a nightmare, I'd imagine.
With exosomes, you can manufacture them, freeze them, and just store them in a standard medical freezer as a stable, off-the-shelf therapeutic product.
So essentially, the exosome is a biological flash drive.
A flash drive, yeah, exactly.
It's this tiny, secure package loaded with therapeutic data and instructions.
Yeah.
But how does it actually deliver that code? Like if
Oh.
it's just floating around in the body, how does it get the message into the damaged tissue?
This is where that lipid bilayer comes into play. Because the exosome is wrapped in the exact same biological membrane material as our own human cells, it acts like a soap bubble merging with another soap bubble.
Oh, I see.
When it reaches a damaged cell, the membranes fuse together and the exosome plugs in, basically downloading its regenerative RNA software directly into the target cell's cytoplasm.
Okay, that makes sense. But if exosomes are just flash drives delivering the instructions, does that mean the computer-like, the whole stem cell itself, is becoming obsolete in modern therapies?
Well, what's fascinating here is that the cell isn't obsolete at all. It's just, shifting roles.
Shifting roles how?
It's moving from being the product to being the factory. The value of the information is in its application.
Right.
The stem cell is still the master computer generating all this incredibly valuable data, but the exosome is simply a far more efficient, safe, and stable courier for that data.
Because it's so small.
Exactly. Because they are so small, these flash drives can penetrate deep into biological barriers, like the blood-brain barrier, that a massive whole cell simply cannot cross.
The factory stays safely in the lab, and we just ship the potent product to the patient.
Yeah.
I love that.
It's brilliant.
So now that we have a solid grip on what these biological flash drives actually are, let's look at the core focus of the research. How the specific tissue source changes the data written on that drive.
Right.
Let's compare the two most common adult sources for these stem cells, bone marrow and adipose tissue, which is just the clinical term for fat.
The proteomic analyses comparing these two are incredibly revealing.
Yeah.
When researchers analyzed the exosomes secreted by bone marrow stem cells, they found massive concentrations of very specific growth factors, most notably fibroblast growth factor 2, or FGF2, alongside bone morphogenetic proteins.
Right, and to translate that into plain English, those specific proteins are essentially the blueprints for osteogenesis bone formation. They also carry factors called rank and rank ankele, which are tied to bone remodeling. So bone marrow exosomes are basically specialized for fracture healing and integrating musculoskeletal tissue.
Exactly.
I like to think of them as the builders. They are bringing the instructions to put up the structural scaffolding, the hard bone.
The builders. That is a highly accurate way to visualize it. Now, contrast that exact same type of vesicle, but harvested from adipose tissue, or fat.
Okay.
The data shows a completely different biochemical signature. Fat-derived exosomes are overwhelmingly enriched in vascular endothelial growth factor A, or VEGF-A.
And VEGF-A is the king of angiogenesis.
Yes, it is.
It is the core signal for building new blood vessels.
Yeah.
So this makes the adipose exosomes superior for wound healing or protecting tissue from ischemia, which is when tissue starts dying because it lacks oxygen.
Right, exactly.
So if bone marrow exosomes are the builders putting up the scaffolding, adipose exosomes are the plumbers laying down the vascular piping to bring blood and nutrients to the site.
The builders and the plumbers. It perfectly captures their physiological roles in the body.
Hold on, let me push back on this for a second, though.
Sure.
If I'm reading the underlying premise correctly, both of these exosomes promote general wound healing.
They do, yeah.
So I'm a bit lost as to why a doctor actually needs to choose between them. If you have a severe injury, like a deep cut, you need both scaffolding and blood flow, right?
Right.
Why go through the trouble of specifically sourcing them from fat versus bone? Couldn't we just use a generic mixed batch for everything?
Well, if we connect this to the bigger picture, we have to look closely at the specific microenvironment of the injury we're trying to treat.
Okay.
Tailoring the therapy is crucial because different tissue environments have vastly different metabolic and signaling needs.
Ah, I see.
Using the builder when you critically need a plumber is highly inefficient, and at the cellular level, it comes down to the specific signaling pathways these exosomes are turning on.
What do you mean by signaling pathways?
Let's look at an ischemic injury, say a severe diabetic foot ulcer.
Ouch.
Okay, the primary issue there isn't a lack of structural scaffolding, it's a catastrophic lack of blood flow.
Right, the tissue is starving.
Exactly, the tissue is starving. Adipose exosomes contain specific cargo that activates what we call the PI3-ACO2 signaling pathway in the patient's existing cells.
Okay, PI3-KECTT sounds like a Wi-Fi password.
Yeah, it does.
In plain English, how does that actually build a blood vessel?
Think of it as a molecular green light. When the adipose exosome plugs into the dying tissue, It flips that PI3-KT switch.
Okay.
That switch tells the local cells to stop dying, start multiplying, and organize themselves into new biological tubes, new blood vessels. It forces the local tissue to sprout piping.
Okay, got it.
Now, contrast that with bone marrow exosomes. They are delivering factors like RANK.
Right, the builders.
Right. RANKL is essentially the signal for the demolition and rebuild crew in your bones. It modulates osteoclasts, which are the cells that clear away old bones so new bone can be laid down. If you deliver bone remodeling signals to a starving diabetic ulcer, it isn't going to save that soft tissue. It's the wrong software for the hardware.
Oh, wow.
The precision of the cargo dictates the success of the therapy.
So the source tissue programs the exosome for its native environment.
Right.
Bone marrow naturally knows how to build bone. Fat naturally knows how to build blood supply.
Exactly.
That is wild. But what happens if the stem cell factory isn't from an adult at all? That brings us to the most fascinating part of this deep dive.
Oh, absolutely.
If bone marrow and adipose represent these specialized adult tissue toolkits, what happens when we look at a primordial source?
Right.
The research details a major proteomic twist when analyzing stem cell exosomes derived from the umbilical cord.
Yeah, the umbilical cord represents a completely different biological paradigm. We are no longer talking about adult tissue, we're talking about fetal-derived tissues, and the exosome cargo heavily reflects that origin environment. An environment that is defined by rapid development, pristine tissue generation, and a total absence of the inflammatory wear and tear we see in adult tissues.
The major discovery in the data was genuinely surprising. They found that while the blood vessel building VEGF and the bone building FGF were present in the umbilical cord exosomes, they were at much lower levels compared to the adult fat and bone marrow.
Right.
But they found something else, transforming growth factor beta, or TGFF. In this specific analysis, TGFF was only detected in the umbilical cord exosome.
Only there, yeah.
It was completely absent in the bone marrow and adipose exosomes.
That exclusivity is a massive finding because TGFS is a master regulator of cellular behavior, particularly when it comes to how tissue repairs itself after trauma.
And the mechanism for how it works is incredible. The presence of TGFF makes these umbilical cord exosomes uniquely capable of inhibiting scar formation.
Yes.
It modulates something called the SMAD pathway. Can you, ELI5 that for us?
Yeah.
How does it actually stop a scar from forming?
Sure. Think of the SMAD pathway as the body's emergency panic button for wound healing.
Okay, panic button.
When you get a deep cut as an adult, your body panics and rushes to seal the breach as fast as possible. It dumps a ton of tough, chaotic collagen into the wound.
It's just to patch it up quickly.
Exactly. That chaotic collagen is what we call a scar. It's fast, but it's messy.
Right.
The TGF from the umbilical exosome essentially intercepts that panic signal. It calms the SMAD pathway down, telling the local skin cells, don't rush, lay the collagen down neatly and perfectly.
Wow.
The result is a repair that looks like the original skin rather than a thick fibrotic scar.
Wow. And it's not just skin. The research highlights they are incredibly effective in joints, protecting cartilage in conditions like osteoarthritis by suppressing destructive factors like MMP13.
Yeah.
And MMP13, if I'm understanding it, acts like microscopic Pac-Man enzymes that just chew up and destroy your knee cartilage over time.
That's a great way to picture it.
The umbilical exosomes go in and turn those Pac-Man off.
Exactly. It is a profoundly powerful, highly protective, regenerative profile.
Here's where it gets really interesting to me. A major fundamental growth factor like TGFA being totally exclusive to the umbilical cord exosomes in this analysis.
Yeah.
Does this mean umbilical cord exosomes are basically the magic bullet of regenerative medicine? Or are they just a highly specialized eraser for taking away scars and joint damage?
This raises an important question about how we view these therapies, and, it's vital we don't think of them as magic.
Right.
It's a direct logical reflection of fetal biology. Think about how a fetus heals inside the womb. It heals entirely without scarring.
Oh, true.
The umbilical cord cells haven't been subjected to decades of oxidative stress, UV radiation, or environmental toxins.
They're fresh.
Exactly.
Oh.
They lack age-related cellular deterioration, a biological concept known as senescence.
So simply put, they aren't tired. They haven't accumulated decades of biological damage and errors like, say, my adult bone marrow cells have.
Precisely. Because they are untouched by aging, they maintain what the literature calls a rejuvenation-associated gene signature.
Rejuvenation-associated gene signature. Got it.
Their exosomal cargo is programmed for perfect, unblemished tissue generation. When you apply umbilical cord exosomes to an adult wound or an arthritic knee, you aren't just giving it raw building materials.
You're delivering a fetal signaling environment.
Exactly. You are chemically instructing the adult tissue to suppress its chaotic scar-forming pathways and instead execute a pristine, scarless repair.
That's amazing.
So, while I wouldn't call them a magic bullet for everything, remember, they aren't as good at building blood vessels as fat exosomes for anti-fibrotic repair and cartilage protection. They are the ultimate mediators.
That is just incredible to think about. The idea that we can literally bottle a fetal healing environment and apply it to a 40-year-old's knee injury.
It really is.
So what does this all mean for you, the person listening right now? Why does this exact mapping of growth factors to tissue origin actually matter for the future of your healthcare?
Well, it points heavily toward the concept of engineerability and nanotheranostics.
Nanotheranostics sounds like a mouthful, but it's essentially the integration of extremely targeted therapeutic delivery with molecular imaging at the nanoscale.
Right.
Because we now understand that the exosome's cargo perfectly reflects the physiological state of the cell it came from, scientists can leverage that.
Yeah.
They aren't just passively collecting these vesicles anymore.
Right. The data highlights that scientists can eventually enrich or intentionally load these vesicles. Because the exosome is a natural, highly penetrative delivery vehicle, scientists can essentially hack the flash drive.
Hack the flash drive.
They can take a bone marrow exosome and pack it with extra therapeutic molecules like specific lab-made messenger RNAs before sending it into the body.
And because we know the baseline profile so well now, we can match the natural strengths of the exosome to the specific disease.
Exactly.
We can use engineered bone marrow exosomes to target osteoporosis, leveraging their natural affinity for bone building. We can use adipose exosomes to treat ischemic strokes or diabetic wounds because of their profound natural ability to trigger blood vessel growth. And we can deploy umbilical cord exosomes for osteoarthritis, utilizing their pristine anti-inflammatory and scar-healing properties.
It really paints a vivid picture of what a pharmacy visit might look like a decade from now.
Imagine you tear your meniscus.
Ouch, yeah.
Instead of getting a prescription for a generic chemical painkiller or facing invasive surgery to cut the tissue out, you get a highly specific engineered batch of umbilical cord-derived exosomes.
Yes.
And it's tailored perfectly to infiltrate your knee joint, plug into your cells, suppress those Pac-Man enzymes eating your cartilage, and heal that tear completely without a single scar.
That is precisely the dawn of personalized, cell-free regenerative medicine. And, I want to encourage everyone to think critically about the broader practical implications of this. By moving away from whole cell therapy and utilizing exosomes, we are completely bypassing massive logistical hurdles. You don't have to perfectly match a donor's immune system to the patient to prevent rejection.
Which is huge.
You have a scalable, safe, and easily administrable biologic that can literally just sit on a shelf. It makes these highly futuristic, highly effective treatments infinitely more accessible to the general public.
It takes it from $1 million bespoke cell transplant procedure down to a vial of biological flash drives.
Exactly.
So to briefly recap the journey we've been on today, exosomes are the ultimate delivery mechanisms, bypassing the risks of whole living cells.
Right.
If you need structural repair, bone marrow brings the bone-building blueprint.
Yeah.
If you need blood flow, adipose tissue brings the vascular-building blueprint. And if you need scarless repair or joint protection, the umbilical cord brings that exclusive fetal anti-scarring blueprint.
It is a perfect summary of how tissue origin dictates therapeutic destiny.
But before we wrap up, I want to leave you with one final mind-bending fact sourced from these papers that we haven't even touched on yet.
Oh, this is a good one.
It's a concept called preconditioning. The research reveals that a stem cell's exosome cargo isn't just static, it is dynamically modulated by cellular stress.
Yes, the cellular environment forces an evolutionary response.
Exactly. If scientists intentionally torture these stem cells in the lab before they harvest the exosomes, say, by subjecting them to severe hypoxia, which is oxygen deprivation, or by inducing thermal stress,
The cells actually panic.
Right, and in their panic, they pack their exosomes with even more potent survival and healing factors than they would under normal, comfortable conditions.
It makes complete biological sense. The cell believes its surrounding tissue is dying, so it floods the local environment with maximum strength emergency regenerative signals.
Just mull that over. We are entering a sci-fi reality where intentionally stressing a cell in a petri dish forces it to create a super healing care package, which we can then extract to heal human disease.
A truly fascinating reality to consider.
The future of healing isn't about giving you a new body part. It's about delivering the exact microscopic message your body needs to heal itself. Thank you for joining us on this deep dive into the microscopic world of exosomes. Keep questioning the world around you, and remember, sometimes the biggest breakthroughs come in the smallest packages.